Welcome to the SANS Science Experience, your source for the most current and impactful research and preprints in science. If you find this deep dive valuable, please do consider liking it and subscribing. It really helps support the SANS Science Experience. Lets us keep bringing you these kinds of critical scientific insights. So today we're digging into something pretty game-changing. It's about atherosclerosis, the root cause of most cardiovascular disease. And this new research, well, it doesn't just pinpoint a surprising new driver, it actually points towards a whole new way to potentially prevent it.
That's right. I mean, for so long, atherosclerosis has been viewed mainly through the lens of, well, cholesterol and those other traditional risk factors we all know about.
Right, lipids.
Exactly. But, the reality is many people still face high cardiovascular risk, or they develop the disease even when they manage those factors pretty well.
Yeah, that's the frustrating part.
It is. And that's precisely why finding completely new targets, things beyond just lipids, is so critical right now.
Okay, so let's unpack this specific discovery. Our deep dive today is focusing on this molecule called Imidazole Propionate, or MP for short.
MP, yeah. And here's where it gets really interesting, like you said. It turns out MP is actually produced by microorganisms, bacteria living right inside our bodies.
Yeah.
In our gut, mainly.
Wow, okay. So not something we eat necessarily, but something our own internal ecosystem makes. That's unexpected.
It absolutely is. And what was really striking in the mouse studies was that just giving them MP, that alone was enough to actually cause atherosclerosis.
Without messing with their cholesterol levels.
Exactly. Even with no changes in their lipid profiles. So that strongly suggests there's a pathway driving this disease that's, well, entirely separate from the cholesterol mechanisms we focused on for decades.
So it's about what our microbes are churning out. And wasn't there something about inflammation too? Did MP trigger that as well?
Yes, that's a key piece. It also sparked both systemic and local inflammation.
Which we know is a major player in hardening arteries.
A critical link, absolutely. Chronic inflammation is like pouring fuel on the fire for atherosclerosis. So MP, triggering it, independently of lipids, points to this really powerful, maybe overlooked mechanism.
Okay, so mice are one thing, but the big question, obviously, is does this hold up in people? What did the human studies show?
Yeah, they looked at that right away. And they found that higher MP levels were strongly linked to how much atherosclerosis people had. This was seen in two separate groups of people, the PESA and IGT studies.
And that link held up even when they accounted for things like cholesterol.
That's the fascinating part. Yes, even after adjusting for all those traditional risk factors, high cholesterol, blood pressure, smoking, you name it, the link was still there.
Yeah.
It really highlights MP as a genuine, independent indicator of atherosclerosis risk.
Right. So thinking practically, what's the potential here? Could measuring MP actually become, a new diagnostic tool, help catch this earlier?
It certainly looks promising. The study showed MP provided additive value. That means it gave extra information on top of the usual biomarkers like LDL-cholesterol or hs-CRP.
So it's not just repeating what we already know.
Exactly. It seems especially useful for spotting active atherosclerosis early on. That could be a huge step forward for, more personalized prevention or treatment. Okay,
this leads to the next big question. If MP is driving it, can we actually block it? What's the mechanism? How does it work? And does this open up new treatment options?
Well, they didn't just find MP. They also figured out how it works. It seems to drive atherosclerosis by latching onto a specific receptor. It's called the Imidazole 1 receptor, or I1R.
I1R, okay.
And this I1R receptor is found on myeloid cells, which are really important immune cells involved in inflammation and plaque buildup.
Got it. So MP hits the I1R on these immune cells, and that kicks off the process, which then leads to the potential therapy.
Precisely. The researchers tested a compound called AGN-192403, and they found that this compound could block that MP-I1R connection.
And did it work? Did it stop the atherosclerosis?
It did, in the mouse models. It prevented atherosclerosis. And interestingly, it worked whether the disease was triggered by giving them MP or by feeding them a high cholesterol diet.
Wow, okay, so it worked in both scenarios.
Yes. And crucially, this blocking effect, the benefit, was completely independent of any changes in cholesterol levels.
So it's a totally different pathway.
A totally different pathway. And it wasn't just prevention, it also reduced the size of existing plaques and lowered inflammation significantly. This really offers a genuinely new approach, potentially for those patients who, despite being on statins and other current treatments, still have a high residual risk of heart attack or stroke.
So, what does this all mean for you listening? Well, here's something to think about. Could targeting the tiny inhabitants of our gut, our microbiome, and the molecules they produce, could that be the next major frontier in fighting heart disease? Especially when our traditional methods aren't quite enough. It's definitely a provocative thought. Remember to like this deep dive if you found it useful, and please subscribe to the SANS Science Experience for more cutting-edge science like this. This has been the Deep Dive, part of the SANS Science Experience.